Hello, in today's video, we shall learn in detail about plant kingdom. By the end of this video, you will have a clear understanding about cryptogams, the seedless plants, which include subkingdom Thallophyta, Bryophyta, Pteridophyta, and Phanerogams, the seed-bearing plants, which include Gymnosperms and Angiosperms, which may be a Monocotyledon or a Dicotyledon. Number of diagrams pertaining to each subtopic have been included throughout the video. Let us begin with the introduction to plant kingdom. Kingdom Plantae consists of multicellular eukaryotes with a distinct nucleus and other membrane-bound organelles. They are autotrophs and utilize chlorophyll pigment to manufacture their own food by photosynthesis. The cell wall is rigid and is composed of cellulose, which provides structural support and protection. They can reproduce asexually by vegetative propagation or by sexual method. Many plants exhibit an alternation of generations in which they possess both haploid gametophytic phase and the diploid sporophytic phase. Let us study about the classification of kingdom Plantae. The kingdom Plantae can be classified into subgroups based on the structure of the plant body, presence or absence of vascular system, flowers, and formation of seeds. They are subdivided into cryptogams, which are seedless plants, and phanerogams, which are seed-bearing plants. Cryptogams are further subdivided into subkingdom Thallophyta, Bryophyta, and Pteridophyta. Phanerogams are divided into Gymnosperms, which are cone-bearing seed plants, and Angiosperms, which are flowering seed plants. Angiosperms with one cotyledon are known as Monocotyledons, and those with two cotyledons are known as Dicotyledons. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us learn about the cryptogams. Cryptogams are a group of lower plants. They are non-flowering and seedless plants with hidden sex organs and reproduce by means of spores. They can reproduce vegetatively by simple fission or fragmentation, asexually by spores, and sexually through the fusion of male and female gametes. Gametes may be isogamous, with both the male and the female gametes of same size, shape, and behavior. Gametes may be anisogamous, when both the male and the female gamete differ in size and behavior, or they may be oogamous, when the female gametes or megagametes are larger and non-motile, while the male gametes or microgametes are smaller and motile. Oogamous type are seen in advanced cryptogams. Plants which show alternation of generations consists of a diploid sporophytic phase and a haploid gametophytic phase. Cryptogams are divided into subkingdom Thallophyta, Bryophyta, and Pteridophyta. Thallophytes contain plant-like organisms which are found in moist places. Their body is thallus-like and lack a well-differentiated body structure. They are mostly autotrophic except fungi, which are heterotrophic. Vascular tissue is absent. Reserve food is mainly stored in the form of starch. Algae and fungi belong to the kingdom Thallophyta. Algae are classified into three classes, namely Chlorophyceae, consisting of green algae, example Chlamydomonas and Spirogyra. Phaeophyceae, consisting of brown algae, example Ectocarpus sargassum. Rhodophyceae, consisting of red algae, example Polysiphonia and Gelidium. Chlorophyceae is a class within the division Thallophyta consisting of the green algae. They may be unicellular like Chlamydomonas or multicellular like Spirogyra. The members of class Chlorophyceae possess an undifferentiated body structure known as thallus. The chloroplast present may be discoid, reticulate, spiral or ribbon-shaped with pigments chlorophyll A and B. The storage bodies pyrenoids located in the chloroplast mainly store starch. Green algae have a rigid cell wall made up of cellulose and pectin. Vegetative reproduction takes place by fragmentation. Asexual reproduction is by flagellated zoospores, and sexual reproduction may be isogamous, anisogamous, or oogamous. Phaeophyceae is a class of brown algae mainly found in marine habitats. They are multicellular and range from simple branched filamentous forms to profusely branched forms. They are autotrophic and possess different pigments like chlorophyll A, chlorophyll C, carotenoids, and xanthophylls. Fucoxanthin is the main xanthophyle pigment which is responsible for their characteristic color. Food is stored as mannitol or laminarin, which are complex carbohydrates. The cell wall consists of inner cellulose for strength and outer gelatinous coating of algin. A root-like structure called the holdfast is present at the base of the algae for anchorage. It possesses a stalk or a stipe and a flattened portion resembling a leaf or a frond which carries out photosynthesis. Brown algae reproduce vegetatively by fragmentation. Asexual reproduction takes place by zoospores, which have a pair of laterally inserted unequal flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Examples of brown algae are Ectocarpus, sargassum, and Fucus. Rhodophyceae consists of red algae. Majority of them are marine. Pigments chlorophyll A, chlorophyll D, phycoerythrin are present in their body. They are mostly multicellular with complex body structure. The food is stored as floridin starch. Cell wall is made up of cellulose, pectin, and esters. Vegetative reproduction takes place by fragmentation. Asexual reproduction is by non-motile spores. Sexual reproduction is oogamous and the gametes are non-motile. Examples of red algae are Polysiphonia, Gelidium, etc. Next, let us learn about the bryophytes. Bryophytes include typically non-vascular plants that live in moist or wet, shady places. Examples are mosses and liverworts. They are known as the amphibians of the plant kingdom because they live in soil but are dependent on water for sexual reproduction. The plant body has root-like, stem-like, and leaf-like structures but do not possess vascular tissue. Bryophytes contain a variety of pigments like chlorophyll A, chlorophyll B, carotenoids like alpha and beta carotene, lutein, etc. These plants show alternation of generations with independent and free-living haploid gametophyte as the dominant and photosynthetic phase in the life cycle. The diploid sporophyte is dependent on the gametophyte for nourishment. Both sexual and asexual reproduction occur in bryophytes. Sexual reproduction takes place by the fusion of male and female gametes, while asexual reproduction can occur through fragmentation, gemme, or protonemal growth. The main plant body of the bryophyte is a haploid gametophyte that produces multicellular sex organs. The male gametangia are the antheridia and the female gametangia are the archegonia. The antheridia are typically stalked, club-shaped structures that produce biflagellate anthrozoids. The archegonia are flower-shaped structures producing eggs. The anthrozoids are released in water and they come in contact with archegonium. Once the anthrozoid fertilizes the egg, a diploid zygote is formed. The zygote develops into a multicellular body called a sporophyte, which remains attached to the gametophyte for nourishment. The sporophyte produces haploid spores by undergoing reduction division. These spores, when released in a suitable environment, germinate to form a new gametophyte. Next are the pteridophytes. Pteridophytes are a group of seedless vascular plants, also known as vascular cryptogams. Example, ferns, Selaginella, equisetum. The main plant body is the diploid sporophyte, which is differentiated into true root, stem, and leaves. The sporophytes bear sporangia on the sporophylls, which produce spores by meiosis. The spores on germination give rise to multicellular gametophytic bodies called prothalli. Antheridia and archegonia develop on the prothallus. The anthrozoids from the antheridium reach the archegonium. Water is essential for fertilization. Fusion of male gamete with the egg present in the archegonium results in the formation of zygote, which ultimately develops into a well-developed sporophyte. Therefore, the life cycle of pteridophytes show alternation of generation between a dominant sporophyte and a reduced independent gametophyte. Next, let us study about Phanerogams. Phanerogams are a group of vascular plants, also known as spermatophytes. They are seed-bearing plants with distinct root, stem and leaves. They are multicellular eukaryotes with well-developed vascular system. They are classified into Gymnosperms and Angiosperms. In Gymnosperms, the ovules are not enclosed by any ovary wall. Example, Cycas and Pinus. The seeds produced are naked. The plant body is a sporophyte with root, stem and leaves. They are woody perennials showing xerophytic characters. They possess taproots, sometimes show fungal association in the form of mycorrhiza, or may be specialized coralloid roots associated with nitrogen-fixing cyanobacteria. Stem is unbranched and the leaves may be foliage leaves or scaly leaves. The reproductive parts of gymnosperms are arranged spirally along an axis to form compact strobili or cones. Male cones have many microsporophylls. Each microsporophyle contains many microsporangia which form microspores or pollen grains. Female cones have many megasporophylls with ovules or megasporangia. The megaspore mother cell divides meiotically, forming 4 megaspores. One of the megaspores develops into a female gametophyte that bears the female sex organs, the archegonia. The pollen grains, when released from the microsporangium, are carried by wind. On reaching the opening of the archegonia, they release the male gametes. Fertilization of male and female gametes gives rise to a zygote. The zygote develops into an embryo and the ovules into seeds. The embryo gives rise to a new sporophyte. Gymnosperms exhibit alternation of generations. Gymnosperms are diplontic because the diploid sporophytes are the predominant phase. Angiosperms are flowering plants which bear flowers and fruits. The diploid sporophyte is differentiated into root, stem and leaves. They are vascular plants possessing true vessels. The flowers are the reproductive organs for the plant. Stamens or microsporophylls and carpels or megasporophylls are arranged in a flower. Each stamen consists of a filament and anther which undergoes meiosis to form haploid pollen grains. Carpel or the pistil consists of stigma, style and ovary. Ovary contains ovules. Through meiosis, ovules produce megaspores which develop into highly reduced female gametophytes known as the embryo sac. Each embryo sac has a 3-celled egg apparatus consisting of 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The matured pollen grains are transferred from the anther to the stigma with the help of various agencies like wind, insects or water. Pollination takes place and pollen grains are transferred to the ovary through the pollen tube. 2 male gametes are released into the embryo sac. One of the male gamete fuses with the egg cell and a zygote is formed. The other male gamete fuses with the polar nuclei and forms a triploid primary endosperm nucleus, a nutritive tissue which supplies nourishment to the embryo. Double fertilization is a unique characteristic of flowering plants. After fertilization, synergids and antipodals degenerate. Finally, ovules develop into seeds and the ovary develops into the fruit. Alternation of generations is exhibited by angiosperms with two distinct phases, the gametophytic phase and the sporophytic phase. Angiosperms are divided into two classes, the dicotyledons and the monocotyledons, based on the number of cotyledons in their embryos. We shall learn about the two classes based on the differences in their features. Monocotyledons are flowering plants with a single cotyledon, while dicotyledons are flowering plants bearing seeds with two cotyledons. Monocots have adventitious or fibrous root system. Dicots have a taproot system. Monocot flowers are usually trimerous, while dicot flowers are usually tetramerous or pentamerous. Calyx and corolla are undifferentiated in monocots. Instead, a perianth is present. In dicots, calyx and corolla are differentiated. In monocots, the pollen grain is monocolpate with a single furrow. In dicots, the pollen grain is tricolpate with 3 or more furrows. Transverse section of the stem of monocots show scattered vascular bundles, while that of dicot stems show vascular bundles arranged in concentric circles. Secondary growth is absent in monocots due to the absence of cambium, while dicots show secondary growth due to the presence of cambium. Leaves show parallel venation in monocots and reticulate venation in dicots. So today we have learnt in detail about the plant kingdom. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.